Hi, welcome to the IB Biology IA overview. Now this is an overview for the new spec, so first exams in 2025, and it's the IA for that because there are some slight differences. So the IA is worth 20% of your overall grade and it's marked out of 24. So if you think about that, if you get 12 out of 24, you're already walking into your IB biology exams with 10% of the grade already. If you get 18 out of 24, you're walking in with 15%. So it's a really, really good opportunity to increase your grades and go into those exams with a really solid foundation. It'll be marked by your class teachers, and just a sample is moderated by the IA to ensure that the marking is fair across the cohort. So I said it's marked out of 24. You'll see at the bottom here that research design is 6 marks, data analysis is 6 marks, the conclusion is 6 marks, and the evaluation is 6 marks, which is slightly different to the old IA. So don't get confused if you see any examples from previous IAs. This is the new marking system. Within the IA, you will choose a research question. You will plan an investigation in order to try and answer that research question. You will collect results in the lab. You will analyse the data. You will draw conclusions from the data, and then you will evaluate the investigation. So pick out kind of what was good and specifically what was bad. Now, when you collect your data, it should represent around about 10 hours of work. So you should have the opportunity to be in the lab for up to 10 hours in order to collect this data. Now we split this so that there is some time for the trial run, so you can check that your method works and make any amendments if you need to, and then we usually do a whole day of data collection, and that generally adds up to about 10 hours. It is possible to do a data-based IA, so this is where you don't collect your own data in the lab and you find some data that somebody else has collected, or you use some data that somebody else has collected. Now this could be a spreadsheet of data collected by your class. So if you've been on an ecology field trip, for example, and you've all collected data in the field and then you've put it onto a spreadsheet, you can use that data. Your school might have spreadsheets from previous years where they've collected data. You can use that. Obviously, you need to make sure that you reference it and credit the people that collected it originally. Or you could use published data. So published data might be things like the incidence of specific disorders or diseases in different countries. It could be linked to something like nutrition or something environmental, but you need to be able to find the data for that relatively easily. And before you start planning a whole IA, you need to make sure that that data is available if you want to do a database IAs. You also may help each other collect data. So if you're not doing a database IA, you can help each other to collect data, but your question has to be original to you. So you can't do the same question and use the same data. If you work with somebody, then your school has to submit their candidate number along with your IA so that the IB are aware of who worked together when collecting data. Because you have to have your own question, there is scope for you to do things like, if you did an ecology IA, you could do the number of snails on the distance from the shore versus the number of snails, and then your friend could do distance from the shore versus the number of starfish, and you could both help to collect that data. That's fine, but your question has to be your question and nobody else's. It's not a group project. The IA is the same for both SL and HL, so I know in some subjects it's slightly different or very different. For biology, it's just the same. The maximum word count is 300 words. So it used to be 12 pages, so if you see that published anywhere, that's the old spec. It's now a maximum of 3,000 words. You can do less than 3,000 words, that's fine, it doesn't have to be bang on, but if you go over 3,000 words, whatever's over won't count, so stick within that word limit. And also any words within charts, diagrams, tables, calculations, titles, or your reference list, the words do not count there. So you can use 3 pages of data, include that in your appendix, and it won't count towards your word count. Okay, what we're going to do is have a look at the different sections and what's required in the different sections. I've also got a few photos within this that might give you some ideas for IAs as we go. So this here is essentially the marking criteria for the research design section. So you can see that it's split into bands, so you can either get 1 or 2 marks, 3 or 4 marks, or 5 or 6 marks. Now, when your teacher's decided which band you're in, they will then decide kind of whether you get 5 or 6 marks or 3 or 4 marks, depending on how strong your work is. So I think that we need to concentrate on the 5 to 6 mark section. So you can see that the research question has to be described within a specific and appropriate context. Now, over here on this side, you can see that the research question should be something along the lines of how does X affect Y? So you want to choose only one independent and one dependent variable, because we want to keep this investigation relatively simple, not too simple, but relatively simple. Once we start throwing in more than one independent variable or dependent variable, it's going to get confusing. And then it talks about the methodological considerations associated with collecting relevant and sufficient data, and then the description of the methodology for collecting or selecting data allows for the investigation to be reproduced. So what you need to have in there is your research question, some background information which gives context to the project, explains the science behind the project, and shows that you have some IB knowledge of this topic, and perhaps in there outline why you've chosen the topic and kind of your interest in that. You used to get marks for personal engagement. You don't anymore, but there is still an element of there has to be some reason as to why you've chosen this project, and your teacher hasn't just given you a specific investigation to do. The method, so we talked about the method in these two. The method should be a numbered list and it should be written by you. If at any point you find yourself copying and pasting a method, then that is not your method. You have to write number 1. And this is what happens in number 1. So you have to be typing up the method. Yes, you can refer to different methods and you can then reference them, but you must write your own method. So it is your method. You need to decide the volumes, the masses, how long something is going to be run for. That's up to you. And within that method, you should also ensure that you explain or describe how you're going to control all of the control variables. So control variables will be outlined before you write the method, and within the method, every control variable should be controlled. There should be part of the method that controls each variable. You also need a risk assessment for your investigation to make sure that what you're doing is ethical and safe within the lab. Here you can see just a couple of pictures. So this one here is beetroot, and I assume that this is probably some sort of alcohol. So that's all about membrane permeability and breaking down the membranes within beetroot in order to release the pigment. So you could do something along those lines. I mean, that's a very standard practical. I wouldn't do that practical, but you could take some elements of that and look at something. We've also got here some chicken. It's possible to do some IAs on kind of tenderness of meat and what you could use to tenderize meat, and then work out a way of measuring how tender the meat is. So just a couple of ideas there. Okay, data analysis. So after you've done your research design, you will write your method and then you will collect your data. You will then present your data. Now, your raw data table should have at least 25 pieces of data within it. So you need to manipulate your independent variable 5 times. So if you've chosen temperature as your independent variable, you should choose at least 5 different temperatures to do within your data collection. And then you need to repeat each trial 5 times. So that will give you 25 bits of data at the very least. Your raw data table should only include raw data, and then the table after that will be your process data. So within your process data table, you will have means and standard deviations. Some projects might have extra data processing, so you might calculate a rate, or you might calculate species richness or species diversity or something along those lines. So there may be additional data processing depending on the title of your project. You'll then have a graph, and the correct type of graph should be chosen, and only processed data should be plotted. So the raw data won't be plotted on the graph because it gets too messy. There'll be too much on there. So we want to see trends, so therefore processed data only. You'll also carry out a statistical test. So you will choose the statistical test and say why you've chosen it. You will carry it out, and then you will interpret it and explain what the results of that statistical test mean. Now, these are the criteria for the data analysis. I'm going to focus on the 5 to 6 section. So you can see that the communication of the recording and processing of the data is both clear and precise. So that's talking about your data tables and making sure that your presentation of the data is clear. We can work out what it is, and it's also correct. So the processing has to be correct as well. The means have to be correct. Standard deviation has to be correct. The recording and processing of data shows evidence of an appropriate consideration of uncertainty. So you need to show the uncertainty of any equipment that you have used. And that's key, because otherwise you won't be able to get 5 or 6 marks in data analysis. And then the processing of data will be relevant to the research question. So your teacher should help you to choose the correct statistical test, and then you will carry it out and interpret it accurately. Here we've got just a picture of a flower growing, a seedling. There are lots of IAs that you could do where you look at how different environments affect the growth of a seedling. So you could look at what you water it with. Like some people in the past have watered with Arabic coffee, have watered with different caffeine solutions, vitamin C solutions, vitamin D solutions. You could look at what you plant the seeds in. So coffee grounds is a classic one. So use coffee grounds, makes it sustainable. Plant them in different kind of potting materials and see how that affects it. We've also got a picture of a beach here, so you could do some sort of transect from the shore further up. A sandy beach probably wouldn't give you very many results because there probably wouldn't be a huge amount of living things in there. But you could use a rocky shore maybe, or a different shoreline, and look at the number of species at different points along that transect or something along those lines. Just another idea. Okay, the conclusion is aiming to answer your original research question. So you posed a question at the beginning, you've collected the data, you've processed the data, and now we're answering that question. Like, did X affect Y? So you also then need to discuss your data. So were there any anomalies? Why were there anomalies? What happened? What could be the reasons for that? Which one had the highest data point, which one had the lowest data point, again, why, are there any reasons, scientific reasons for that, why were there differences in standard deviations, if there were, like what could have caused a higher standard deviation and a lower standard deviation. Your conclusion should match with the trend on your graph. And you can also discuss the results of the statistical test in more detail here. So you can talk about whether the results of the stats test match up with the graph that matches up with the data table, etc., etc. You also, if you have a look here, you can see the conclusion is justified and relevant to the research question, fully consistent with the analysis present. So it does have to actually marry up with your data. And then it says the conclusion is justified through relevant comparison to the accepted scientific context. So you have to find some similar studies, some similar data to your investigation, see what they got and discuss your results in comparison with theirs. So did your results match up with their results, and why could that be? Did your results not match up with their results, and why could that be? Now, often students say that they can't find any similar studies, but it doesn't have to be the exact same study. It could be the same independent variable, or it could be the same dependent variable. If there's absolutely nothing and your research was really, really original, then you can just use actual science to back up your results and discuss it, saying it happened because of, and then discuss why it happened using your scientific knowledge. Up here we've got a picture of different fruits with bananas. So bananas release a gas called ethylene, and that then causes the ripening of other fruits and vegetables. This is a bit of a classic IA that comes up every year, I find. You can do something with that. You can amend it, make it your own. Maybe you can look at like mass of banana peel. Does it have to be a whole banana? Could it be a peeled banana, etc.? Things like that. And then this just measures the oxygen saturation of the blood and the pulse. I mean, oxygen saturation for everyone should be like 98, 99%. And but obviously pulse can change, so you might want to do some sort of exercise IA. In the past, people have done things about like watching different genres of film and then looking at pulse, and that doesn't work particularly well, but you could do something around that. But yeah, heart rate is a really nice, easy one to measure. And then finally, we've got the evaluation. So the evaluation is going to outline the weaknesses of the investigation. So it used to be strengths and weaknesses. Now they've taken strengths out. If you have a look here, the report explains the relative impact of scientific methodological weaknesses or limitations, and then realistic improvements that are relevant are explained. So I'd go with at least 5 weaknesses, and the weaknesses need to be like real weaknesses. So for example, you could say that looking at the distance that a bubble moves when a gas is released would be less accurate than using a gas syringe to measure the volume of gas. So you want to think of real weaknesses that you could then improve. Now, when you're writing your IA, if you think, oh, actually, I could use something else, but we don't have that equipment, maybe I should do a different IA. No, make a note, like do your IA, but make a note of that as a weakness, and you can then gain credit for it within the evaluation. So just as you're going through, if you think about weaknesses, it might be a good idea just to write them down so you have them at the end. You need to correctly refer to increase in reliability, accuracy, and precision. So lots of students will just write, and this will make it more accurate. But you need to understand that reliability can be improved by collecting more data. Accuracy can be improved by using better equipment. So for example, the gas syringe, rather than looking at how far a bubble moves. And then precision might be able to be improved by removing human error or getting the same person to collect all of the results or something along those lines. Each weakness should be described, its impact should be explained, and a suggested improvement outlined. Now each weakness should be a fairly hefty paragraph. It's not just a couple of lines, it should be a decent paragraph. Now, I always find that the evaluation is the worst bit of an IA and students score the lowest. And the reason for that, I believe, is that at this point, the students are bored of doing the IA and they've got like 5 other IAs to do. So make sure that you stay on top of it and that your evaluation is as good as your research design when you were really into it and you were like, this is going to be the best IA ever. Make sure that that continues and the evaluation is good, because it is typically the worst area, the worst scored area. Now here we've got someone using a quadrat, so you could do some sort of ecology IA whereby you randomly select areas within a field and you count the number of a specific species there. It could be percentage cover in the quadrat, but that's a nice kind of relatively simple but good results kind of IA. And then this one here is a VR headset. I've never had anyone do any VR based IAs, but I just feel like there's probably lots of ideas whereby you could use VR and you could maybe look at I don't know, heart rate in different types of VR games or something along those lines. I just thought it probably lends itself quite nicely to IAs. And that's it. That's everything you need to do for your IA.